In this video, I'll be talking about conjugation, specifically looking at the PVJT128 plasmid and how it's transferred from a donor E. coli cell to a recipient E. coli cell. This is a diagram of our E. coli donor cell, which, in addition to the normal parts of a bacterial cell, also contains plasmids, indicated by the red circles. These plasmids are respectively called the helper plasmid and PVJT128, which is named after Valerie J. Thompson. Keep in mind that these plasmids are double-stranded DNA. There are several important sequences on the PVJT128 plasmid, including the origin of transfer, also known as ORET, lac operon, TMP gene, chloramphenicol resistance gene, and kanamycin resistance gene. For the conjugation part of the lab, the ORET and chloramphenicol resistance gene are the main focus. On the helper plasmid, the transfer or TRA genes are necessary for bacterial conjugation. The plasmid helps because the TRA genes encode important proteins like the nicking enzyme. This enzyme recognizes PVJT128's origin of transfer region and nicks the DNA strand on which the ORET resides, creating one single-stranded DNA that is cut and one that remains circular. The TRA genes also encode proteins necessary for forming the pilus and transferring the cut single-stranded DNA to the recipient cell, which we'll see right here. On the left is the donor cell, now with a cut single-stranded DNA plasmid and the remaining single-stranded circle. On the right is an E. coli recipient cell that doesn't currently have any plasmids, but it has a nalidixic acid resistance gene in its chromosome, unlike the donor cell. The proteins made from the TRA genes in the donor help make a pilus, or mating bridge, between the donor and the recipient, and these proteins also assist the single-stranded PVJT128 DNA into and across the pilus. When the single-stranded DNA makes it into the recipient, it circularizes, and the pilus is degraded. In both the donor and the recipient, the single-stranded DNA is copied so that the plasmids become double-stranded and fully functional. The experimental setup for conjugation is elegantly simple. All that's needed are Eppendorf tubes, pipettes, a Bunsen burner, a vortex, a hot water bath, agar plates, saline, and of course, the bacterial E. coli strains. We have donor and recipient cells contained within their own Eppendorf tubes. We mix 500 microliters from each tube into what's called the mating tube. The mating tube is put in a hot water bath for an hour to facilitate the interaction and conjugation between donor and recipient cells. We remove the mating tube from the bath and make dilutions of its contents to be plated on a special agar. This agar contains chloramphenicol and nalidixic acid. Only cells that are resistant to both antibiotics will survive and grow on the plates. In other words, only recipient cells that have received the plasmid PVJT128 will grow on these plates. These cells represent successful conjugation. The 10⁻⁴ dilution can be taken directly from the mating tube and plated onto the agar. The other dilutions must be made through the serial dilution process. For example, 100 microliters of the mating tube is added to 900 microliters of saline and vortexed to create the 10⁻⁻1 dilution. To make the 10⁻⁻2 dilution, 100 microliters of the 10⁻⁻1 solution is added to 900 microliters of saline and vortexed. These dilutions are kept in their own Eppendorf tubes, and we plate 100 microliters of each solution onto their respective plates. A similar process is done for the original donor and recipient strains, but different kinds of agar plates are used. The donor cells are plated on nalidixic acid agar to check that it is not resistant and therefore should not grow, unless some cells undergo spontaneous reversions that suddenly make it resistant. A similar test is done for recipient cells on chloramphenicol agar. We have to check for spontaneous reversions in both the donor and recipient cells because they would skew our calculations for percent conjugation. Measuring the amount of spontaneous reversions, if any, allows us to adjust our percent conjugation calculation. We also grow recipient cells on nutrient agar, which is a hospitable environment for growth. The colonies that grow on these plates allow us to calculate the total number of recipient cells that were actually in the mating tube. After solutions are plated, we let the bacteria grow. Once the bacteria grows on each of the plates, we count the number of colonies we see on each plate, not including satellite colonies. To find the number of cells, we divide the number of colonies by the product of the plating factor and the dilution factor. These factors represent the fraction of the original aliquot used. Percent conjugation is calculated using this equation. the number of recipient cells with plasmid, minus any spontaneous reversions, divided by the number of recipient cells in the mating tube. Next time we'll talk about part 2 of the experiment, transposition mutagenesis.